RNAct, enabling proteins for synthetic biology and bioanalytics.
Hello, I am Joel Roca, and today, as part of the RNAct webinar series, I will give you a general introduction to protein design. It's a field that benefits from both experimental and computational methods, where the main goal is to modify or build a protein from scratch so it can adopt a specific 3D structure. This is what we call the protein fold. Just as a curiosity here, the tin barrel that you can see as an O in the protein world is the first successful de novo design of this kind of fold after 25 years of efforts. It was carried out by David Baker's group, and I think it clearly exemplifies how complex this whole procedure is. In its simplest form, proteins are just a chain of letters. This is what we call the amino acid sequence. And encoded in this sequence is the way that protein can fold. And when it folds, it can adopt different secondary structures, like beta strands, but also alpha helices, and those secondary structure elements are connected by the loop regions. The fold that the protein has, so this 3D structure that you can see here, is what will ultimately lead to that protein to accomplish a specific function. And there are many of them. We have, for example, the antibodies, like the immunoglobulins, bind to specific foreign particles such as viruses or bacteria to trigger immune response, protecting this way our bodies. Then we also have the proteins that work as enzymes, transforming a set of one or several substrates into a set of products. We could find in this category, for example, the kinases that transfer phosphate groups from higher energy molecules like ATP to other substrates that can be other proteins, for example, regulating this way different biological reactions. Then we also have the messenger proteins, such as some hormones that transmit signals around the body for coordinating different biological processes. Then we have some proteins that have a structural role, like the actin and myosin fibers that are responsible of the contraction process in the muscles. Finally, one last example would be the transport proteins, like the transmembrane proteins that carry different ions or molecules across the cell membranes. So the goal of protein design is to find new sequences that fold into a specific 3D structure and sometimes also link that to specific function. It is a relatively new field of structural biology, and it was originally conceived as just the generation of new proteins completely from scratch, from zero. But nowadays it has a broader definition. We're going to talk in this webinar about the different approaches that researchers are taking to create new proteins. But we will start with a bit of history. Since the discovery of proteins back in the 19th century and their basic building blocks, the amino acids, it was long thought that protein design, it was impossible, that it was something that only nature could do. But it was already some 40 years ago, in the 80s, where some researchers started taking the first steps. And purely based on physical models, they were able to manually design and synthesize small peptides from scratch. During the 90s, the advance of computers allowed the introduction to computational design guided by physicochemical principles. This allowed to design bigger proteins and with a better success rate. And that means that from the proposed designs that they could find, they were more that at the end were experimentally assessed and successful. This led to the first computationally designed completely novel protein. That you can see here are these alpha helices packed against each other that was experimentally verified, and this experiment was carried out by Walsh and colleagues back in 1999. During the coming years, the increasing availability of more and more protein structures, essentially crystals at the moment, enabled the fragment-based strategies, that is, taking different bits of many different proteins and combine them to create new ones. So at the end you were creating structures that nature had never come up with. Nowadays the field is evolving so fast is difficult to keep up. The increasing number of computational tools for protein design or for protein structure predictors such as Rosetta Fold, AlphaFold, and so on is making this whole process easier than it has ever been. But at the same time, it's opening many new challenges, and there is still, of course, the protein design is not trivial and still very far from it. And there are some fields that now are that are now getting more effort, let's say, like the functional design, that is designing proteins that can accomplish certain functions, or also designing protein assemblies. Those, I could say, the most hot topics at the moment in the protein design field. Currently, we can talk about two main ways of approaching this protein design process. This is the template-based design in one side, where we use known proteins to build new ones, and on the other side we have the de novo design, when either the sequence or the sequence, the structure, or both are completely unknown beforehand. When using the template-based design, we're going to start with this approach. The aim is to define or modify an amino acid sequence from already known proteins, so that will adopt a stable and functional three-dimensional structure. And to accomplish this, there are different methods that have been used and that has been proved successful in the last years. We have, for example, the modular design. This is one of the most common strategies, and in this case, those are proteins which consist of a single subdomain module of secondary structures, different alpha helices or beta strands, repeated several times. Essentially, it's the same amino acid sequence block, or almost the same, that is copied one after the other, so you can get a bigger structure when it gets associated. This can be done using either identical or different modules. Then, a bit slightly more complex, they try to introduce symmetry in the structure, such as some natural proteins like tin barrels. With this strategy, you can achieve globular folds that you cannot create with the other method, where the repetition of these subdomains will finally generate this internal symmetry of the protein. These ones present a higher structural complexity than the linear repeats, because in those cases the internal packing is crucial for the stability of the protein. You really need to create here this hydrophobic core, so the residues can interact with each other and stabilize the core of the protein, and then you should have like some polar residues exposed to the solvent to keep, this globular fold. A clear example of this symmetrical design was the first protein designed with a tin barrel fold. It's the one I mentioned in the first slide, carried out by David Baker's group back in 2016. Another way, even more challenging, is to design a protein that can accomplish a specific function. Since there are many structural scaffolds that can tolerate one to several modifications on its sequence without jeopardizing it, it's possible to modify natural proteins and alter their functions, improving them, for instance, or the other way around, blocking them. One of the most common examples is the design of antibodies that can not only bind to antigens, but also perform catalysis of different chemical reactions. Another popular example is the incorporation of binding sites into a natural protein, or modifications on small molecules that can become inhibitors on protein-protein interactions, for instance. Some of the most common ways to perform this small alterations is by doing experimental in vitro mutagenesis. That is done by mutating the DNA sequences of the parts of the protein we want to express, and then using an expression system like bacteria, for example, to generate many copies of slightly different proteins, so we can finally assess that the functions of those, so which mutations are interesting for the goal of the specific process, the specific project. The combination between for this process between the computational design and this high throughput screening that uses this random mutagenesis quite often, it's giving very successful results, and it clearly helps to get a better picture and to see the landscape and the connection between the sequence that we have of the protein and the function that it can ultimately accomplish. Finally, molecular dynamic simulations have proven very helpful to improve the protein design cycle. This approach provides useful information about the stability of the folded proteins, which is crucial to achieve the desired protein function. But it can also help to assess how stable the stability of the protein by checking the unfolding, for instance. So if you design a new protein, but then in the molecular dynamic simulation you quickly see that the protein just unfolds, even you have a folded initial point, it probably shows that you don't have enough interactions in the core of the protein to keep that structure stable. Simulations are used to screen different protein candidates these days, so you do some kind of filtering step before the final experimental tests that usually are way more time consuming and expensive. The problem of the limitation of the template-based design is that we are restricted to the proteins that have already been observed in nature. The number of natural proteins is in the order of 10 to the power of 12, more or less. But if we take, for instance, a sequence of 100 residues, because we have 20 amino acids, the number of possibilities of that sequence is 20 to the power of 100. So you can see that there is a huge gap between the part that the nature has explored and what could be theoretically possible. So this is where the novel design comes in. Here the researchers just want to build a protein completely from scratch. That is, there are different approaches, as I'm going to show, but on one side we have the cases where you know the sequence of the protein, so you just make a sequence, but you don't know into which kind of fold is gonna adopt. And this part is called the structure prediction, and it's also part of this. It's also engloved in this de novo design process. In those cases, the only thing we need to do is to of course it's not trivial, but the step that must be taken is to predict the backbone first from that sequence we have available, and then sample the different rotamers and try to find the structures that minimizes the energy, so the one that is more stable. Now it's of course worth mentioning that during the last couple of years there have been several high accuracy predictors that has helped this process a lot. It's been a huge leap forward, like AlphaFold and Rosetta Fold, and this year in the last CASP assessment there has also been few very good new structure predictors that I think are worth taking a look at, that definitely have improved a lot, have helped a lot in the field, especially with rigid structures, but we still have all the remaining intrinsically disordered structures, for instance, so parts that do not have a clear fault that still remain a challenge. Then we have the opposite case. When we know the fault that we want to have, the 3D structure, but we don't know which sequence fits that fault. In those cases, this approach is called fixed backbone design, and the scaffold of the protein is clear, but we need to elucidate the sequence by finding the different rotamers and different combinations of residues that satisfy that structure and that minimizes the energy again. Finally, we have the case with the highest number of degrees of freedom, that is when we don't know either the sequence, either the structure. And for that, usually researchers took three main steps. First, they sample backbone conformations. So they start the design process. When you start the protein design from scratch, this is what is called the pure de novo design. The first step is to define a large set of backbones, usually in the order of thousands. This can be made by either assembling different small peptide fragments, or also just by using algebraic equations to specify the geometry of the different parts of the protein. The second step then is for each of the design backbones, to find a sequence that fits into that structure with the lowest possible energy. This energy considers all the forces related with the protein folding process, like the variable of hydrophobic residues, as I've mentioned before in the globular folds, also the van der Waals forces and the intraprotein hydrogen bonds, among other interactions. So any kind of interaction is important at the end to keep the protein stable and well folded. Once the sequence is defined, there is an important last checkpoint, that is to validate that the lowest energy state of the design sequence folds into the design structure. So essentially that there is that both sequence and structure converge together. This is known as the ab initio structure prediction, and only if the results of this computational test are in good agreement, then the protein will be experimentally characterized. As you can imagine, this whole process is far from trivial and computationally very expensive, but has allowed the design of many proteins, many de novo proteins in the past few years. The results can then be checked either with X-ray crystallography or nuclear magnetic resonance. And looking at the obtained structures, and then you check the structures that you solve if they are in atomic agreement with the proposed designs. Although there are still many challenges to overcome in the field, one of the main limitations of this process, it's quite simple actually, is that many of those proteins cannot be expressed. They might be toxic for the bacteria, for the expression system that the researchers might be using, or they will just not express properly, will just not fold. So, as I just said, this protein design is still far from trivial, and there are many work in the field to improve all those steps, and also to, as I mentioned before, also to confer those proteins new functionalities. Like there are now there are quite some papers out, but there was a very interesting one about kinase switches, also so protein switches is using kinases, also from David Baker's group, that I think is very interesting. So I could say that the field is very alive. It's worth trying to keep up, because there are many things coming soon that will be definitely very interesting in order to have new applications and to keep using proteins for new vaccines, for many different applications. And I think that would be all from my side for this protein design general introduction. I will also use the occasion to invite you to watch the other webinars on the RNAct YouTube channel. In case you're interested, you can see them listed here. You can also find the different references I used to prepare this webinar. And also thank, of course, Dr. Ana Pérez-Raffles for contributing to the preparation of the webinar. And finally, the European Union and RNAct for the funding, and the Bio2Byte group and my university, VUB, for hosting me and for taking so good care of me. So, and finally, thanks all of you for watching. Cheers!